The next speaker is Dr. Manoj Mahajan from Udaipur, and he'll be talking to us today on first line Osimertinib for previously untreated patients with NSCLC and uncommon EGFR mutations, the Unicorn phase 2 study. So over to you, Manoj.
Thank you, sir. Good morning, everyone. So I'll be talking about the unventured territory, probably not very good study, but still study results, I'll say. But these kind of studies are needed for our better understanding of the drugs. So this drug evaluated Osimertinib as first-line treatment for NSCLC with uncommon EGFR mutations. So if we look at overall incidences of all other mutations, most of the mutation, especially exon 19 deletion, L858R, these constitute nearly 70% of most of the patients, especially in Indian context. So remaining 30% are uncommon EGFR mutations, which are 14% are exclusive single mutations, and around 14% are compound mutation, 14-15%. So they will have two mutations simultaneously. That is why imparting a resistance. So if we look at the overall uncommon mutations, which are the uncommon mutations? Amongst the uncommon mutations, the most common ones are G719X exon 18, S768I exon 20, L861Q exon 21. So the study intends to evaluate the effect of Osimertinib in such kind of mutations. As we know, first line and Geftinib and Erlotinib have resistance to such kind of mutations. So what are the effects of Osimertinib in this setting that study is intending to do? So I have given the background. The main challenges are like uncommon mutations have limited treatment data and show poor responses to first generation EGFR TKI. We have the second generation TKI, Afatinib, which has pooled analysis of Lux-Lung trials and probably still a standard of care, and even after this results, we'll see at the end, we'll conclude what would be the better option. So trial design, we have discussed. Enrollment period from 20 to 22, very limited number of patients, 40 eligible patients from 32 hospitals in Japan. Study dose was standard, Osimertinib 80 mg once a day, minimum 6 months follow after enrollment was done. So primary endpoint was overall response rate, secondary endpoint PFS and disease control rate, and duration of response. Participants, nearly 42 patients enrolled, but finally 40% patients were eligible. Inclusion criteria, histologically confirmed metastatic NSCLC, uncommon EGFR mutation as we had defined. It can be either PCR by NGS, ECOG PS 01, 0 to 2, and at least one measurable lesion as per the RECIST criteria. Exclusion criteria, symptomatic brain mets were excluded, asymptomatic were included. Other previous treatment with EGFR TK was excluded, so it was treatment naive patients. Other significant medical conditions are active infections. So doses and follow-up we had discussed. So this is the patients, 40 patients enrolled, and finally 40 patients completed the study and evaluated. So if you look at the demographics, nearly half were male, half female, 92.5% were adenocarcinoma, nearly 40% were never smokers, 50% were former smokers, and around 7.5 current smokers. Amongst the uncommon mutation, the most common findings were G79X, 50% of the patients, 20 patients, S768I, 25% of patients, L861Q, 20% of patients. And compound mutation, nearly 45% patient had compound mutations, which involved combination of uncommon EGFR mutations. And mutation detected by NGS nearly 40%, PCR based nearly 60%. So these are the study characteristics which we discussed. Coming to the results, overall response rates nearly 55% in all patients. If we look at the solitary mutations, only 45% overall response rates. Compound mutations had slightly higher mutations rate as compared to solitary mutations. And if we go mutation specific, so the least common L861 had a higher overall response rate to the tune of 75%, but that may be a limiting factor because of number of patients, and other has 45 to 50%. So if you look at the PFS, median PFS was around 9.4 months. Solitary mutations, median PFS 5.4 months. Compound mutations, median PFS of 9.8 months. Patients with asymptomatic brain mets had a shorter median PFS, 4.5 months, compared to those without, around 9.8 months. So these are the median PFS graphs. Clinical activity is there in L861 more. We can see a longer follow-up for the rarer mutations, but again, its studies is limited by number of patients recruited. Common side effects were serious adverse events, mainly ILD, again 12.5%, cardiac events nearly 2.5%. Dose reduction required in around 7.5% of the patients. So this is the same data. Efficacy in the brain mets, as we have discussed, median PFS with brain mets 4.5 months, without brain mets 9.8 months. So if we compare with the other EGFR TKIs, our current standard of care, osimertinib shows superior efficacy as compared to the first generation TKIs, overall response rate with 55%. If we compare with the second generation TKI, afatinib has the response rates up to the 70% and median PFS of almost more than 12 months. So not a statistical design study, but afatinib is still showing the better results as compared to the osimertinib in uncommon mutations. So to conclude, a major limitation of this study was very small sample size, ethnic homogeneity, only Japanese patients were included, heterogeneous mutations, resistance mechanism not explored due to insufficient tumor samples, no phase 3 comparison, and CNF efficacy. We need further studies. Thank you.
Thank you, Manoj.